Monohybrid Cross. Objective: to understand the inheritance mechanism of a monohybrid cross. A monohybrid cross is a cross between two parents that breed true for different forms of a single trait. During the 1860s, Gregor Mendel, an Austrian monk, proposed the basic rules of inheritance. He began his experimentation with pea plants that bred true for a single characteristic, seed color. Because there is only one characteristic which is being bred, it is known as a monohybrid cross. Mendel bred peas with yellow seed coats for a few generations to ensure plants in subsequent generations would have yellow seeds. Similarly, he self-pollinated pea plants with green seed coloring, so all plants in subsequent generations would produce a green seed color. He then cross-pollinated the parental plants, sometimes referred to as the parental generation, of yellow seeds with the plants of green seeds. In the F1 or filial generation, each of the plants yielded seeds with yellow color. Next, Mendel crossed the seeds from the F1 generation to produce the second filial or F2 generation. In the F2 generation, the plants with yellow seeds outnumbered green seed plants 3-1. Now, let's examine the inheritance mechanism of both phenotypes, which are the visible external characteristics, and genotypes, which is the cell's genetic makeup. Each trait is controlled by a pair of hereditary units we now refer to as genes, with one unit originating from each parent. The alleles, more commonly known as forms, of the gene for seed color would be the same in true-bred plants. These will be represented by YY and YY for the parent plants. Crossbreeding YY with YY plants produces YY plants in the F1 generation. This can be illustrated through the use of the Punnett square, a diagram which is used to predict all gene combinations in a cross between two parents. The F1 plants receive a Y allele from the yellow seed parent and a Y allele from the green seed parent. Although the traits for both colors are in the genes, the plants will exhibit only one characteristic, the yellow seed color. The F1 plants have yellow seeds because the Y allele is the expressed trait, meaning it will dominate over the Y allele. Thus, the Y or yellow colored seed allele is referred to as dominant, while Y or green colored seed alleles are referred to as recessive. The gametes from the F1 generation with a heterozygous genotype are matched in the Punnett square. Now let's look at the F2 generation using the Punnett square and observe the changes that occur. The square reveals that the F2 generation actually yields three kinds of plants, unlike F1 which only had the one type. In the F2 generation, 1/4 of the plants has a homozygous yellow seed color and will produce yellow seeds. 1/4 of the genotypes show a homozygous recessive nature, that is, both of the traits for green seeds. These plants will bear green-colored seeds. The remaining one half of the genotype shows a heterozygous nature, as these will have both the dominant and recessive traits. The results let us see that 3/4 of the plants have a dominant yellow trait and, as such, will produce yellow seeds, and 1/4 of the plants have a recessive green trait and, as such, will produce green seeds. Thus, the Mendelian ratio for a monohybrid cross is 3:1 when looking at phenotype and 1:2:1 when looking at genotype. Summary: a monohybrid cross is based on a single trait with two alleles. The parents of a monohybrid cross are true-bred. All of the first, or F1, generation offspring will exhibit the same single trait. In the F2 generation, the progeny exhibits a 3:1 ratio of the trait.